Have you ever wondered how a simple plant, something that's been used in folk medicine for centuries, actually becomes a modern, life-saving pill? Well, it's a fascinating journey that starts out in a field and might end up in your local pharmacy. Today, we're going to follow that exact scientific path, from plant to prescription. In that idea of a bridge, it's absolutely perfect, because that's what this process is all about. It's the link between ancient wisdom, what our ancestors figured out through trial and error, and the super rigorous science of modern medicine. That bridge has a name: biological screening, and it's how scientists systematically hunt for the active ingredients that nature has hidden away. So this really gets us to the heart of it, right? I mean, it's one thing for tradition to say a plant works, but how do scientists prove it? What are the exact, meticulous steps they take to turn a simple root or a leaf into a pure, potent, and proven medicine? That's what we're here to find out. Okay, so here's our game plan. We're going to follow the entire scientific pipeline, and you can think of it like a giant funnel. We're going to start really wide with all of nature's possibilities, and then we'll systematically narrow it down. We'll go from finding the right plants to getting that first exciting hit, then isolating the single active compound, testing it in living systems, and finally, we'll see how new tech is making this whole thing faster than ever before. So let's start right at the top of that funnel. Before we can even think about picking a plant, we really need to get our heads around the fundamental process that drives this whole field: biological screening. So what does that actually mean? At its core, biological screening is basically a methodical hunt. This is not about random guessing at all. It's a really systematic process for testing countless natural compounds to find what scientists call hits. And a hit is just that first little glimmer of promise, that first sign that a plant extract might, just might, contain a future medicine. Okay, so now we're at the widest part of our discovery funnel. It's time to cast the net. And you have to imagine, with millions of plant species on Earth, scientists face this huge challenge right from the get-go. Where do you even begin to look? Well, scientists basically have two main strategies. On the one hand, you have the traditional approach. This is where you follow the clues left behind by history, focusing on plants that people have been using for ages. This is kind of the safe bet. It gives you a much higher hit rate. But on the other hand, you can take a pure shot in the dark. You can randomly screen plants from all sorts of ecosystems. The odds are way lower, for sure. But a discovery here, it could be a total game changer, a completely new kind of medicine the world has never seen. So once you've picked a plant, you've got to get the good stuff out. Just think about making a cup of tea. You use hot water, right? That's a polar solvent, and it's great at pulling out water-soluble things like tannins. But what if the magic ingredient is an oil or a wax? Well, those are non-polar, so water won't touch them. You'd need a totally different solvent, maybe something like hexane, to dissolve it. Choosing the right tool for the job here is absolutely critical. All right, we have our extracts. Now the real filtering begins. This is where thousands of samples start flooding into the lab for their first round of tests, and they're all designed to answer one simple question: does anything in here show a little spark of interesting biological activity? Now, this first round of screening is all done in vitro, which literally just means in glass. So yeah, we're talking petri dishes, test tubes, that kind of thing. The goal here is all about speed and scale. You want to test thousands of samples quickly and cheaply. We're not ready for a living organism yet. We're just looking for a basic chemical signal that says, hey, something interesting is happening over here. I just love how elegantly simple some of these tests are. Like, to see if an extract can fight bacteria, you just soak a little paper disc in it, pop it on a petri dish that's covered in microbes, and you wait. If the extract works, you get this amazing clear circle where the bacteria just can't grow. That's your zone of inhibition, and a bigger zone means you've probably found something pretty powerful. And here's another brilliant one. To find antioxidants, scientists use this really vibrant purple chemical. When they add an extract that has antioxidants in it, it neutralizes the chemical and poof, the whole solution changes from purple to yellow, just like that. It's almost like a magic trick, a simple visual clue that tells you you've found what you're looking for. So how do scientists measure how well something works? Well, a crucial number here is called the IC50 value. This is basically the concentration you need to get 50% of the effect you're looking for. Now, this is a little counterintuitive, so listen up. You actually want a low number. The less stuff you need to get the job done, the more potent and active your compound is. So lower is better. Okay, so we've found a promising extract. Now we enter the next stage, and this is where the real detective work begins. See, our crude extract showed promise, but it's a messy soup of hundreds, maybe thousands of different chemicals. The mission now is to find the single culprit, the one molecule actually responsible for the cool stuff we saw. Luckily, scientists aren't just flying blind. Those early tests give them what you could call a chemical fingerprint of the extract. It tells them what general families of compounds are in there. Things like flavonoids, which are often antioxidants, or alkaloids, which is the group that includes caffeine and morphine. This fingerprint is like a crucial clue that guides the entire investigation. Now, the main technique for doing this is called bioassay guided fractionation. I know it sounds super technical, right? But the idea is actually pretty simple. It's just a loop you repeat over and over. You separate your mixture into a few simpler parts, you test each part, you throw away the ones that don't do anything, and you keep the one that works. Then you take that active part, you separate it again, and you just keep repeating the process until you're left with one single pure compound. And we've done it. We've managed to isolate one single pure compound that works wonders in a petri dish. But now comes the giant leap. Does it actually work inside a complex, living creature? This is the ultimate test before we can even begin to think about trying it in humans. And this really shows you the key difference. The in vitro or in-glass tests are simple. They're controlled, they're fast. But the in vivo tests, the ones in living animal models, well, they're slower and way more complex. But they are the only way to answer the really critical questions about how effective it is, if it's safe, and how a body actually handles the compound. You know, how it's absorbed, used, and eventually gotten rid of. These are the absolute make or break questions. Does it actually work? What's a safe dose? And at what point does it become toxic? And then there's its pharmacokinetics. That's a fancy term for how the body absorbs the drug, moves it around, and eventually gets rid of it. Without solid answers to these questions, a compound's journey just stops right here. This entire process, this whole funnel we've been talking about, from picking a plant to purifying a compound, it used to take decades. I mean, a whole career could be spent on one molecule. But today, technology is supercharging every single step of the process. You know, we're living through a total revolution in drug discovery. The slow, painstaking, manual lab work of the past, it's quickly giving way to robotics, artificial intelligence, and incredibly powerful analytical tools that are making the entire pipeline faster, cheaper, and a whole lot smarter. I mean, just look at some of these tools. We've got high-throughput screening, which uses robots to run thousands of tests a day instead of just a few dozen. We've got virtual screening, where AI can predict how millions of compounds will work on a computer before anyone even touches a test tube. And advanced techniques that can separate and identify all the chemicals in a mixture in one single, super fast run. It is a whole new ballgame. So we followed the entire journey, from a simple plant to a potential pill, all the way across that bridge from ancient knowledge to modern medicine. But when you think about all this new technology accelerating the search and the fact that there are millions of species still completely unexplored, it leaves you with one incredible question. What amazing cure is out there, right now, just waiting to be found?